Now, I'm going to ask Professor John Pickett to come up to the stage to introduce our next speaker. John?
Thank you, Maurice. It's a great privilege and pleasure to introduce the next speaker and the subject, indeed. I think there's a tremendous amount of general knowledge around on the fact that we need fertilizers to produce anything like the amount of food that we're producing at the moment, and yet we've heard very eloquently described the needs for the future. Few people, though, know quite how we do make, certainly, nitrogen fertilizers. The Haber-Bosch process is a wonderful piece of chemical engineering, but it's not understood, and it's not understood in the sense that it's not sustainable. We've really got to do better. We've got to stop using such a vast amount of fossil fuel in creating our fixed nitrogen. I know it's a sophisticated process that's becoming more diverse and that there are new approaches, but they all involve non-sustainable production of fixed nitrogen. So it's biological nitrogen that we have to concentrate our attention on for the future, for sustainability, and for achieving the kind of goals that we have before us. The next speaker then on this subject, good gracious me, is educated in terms of his doctorate at Berkeley, California University, and he obtained that in 1998. And then he moved from plant pathogen interactions to Stanford University, where with Sharon Long he started his work on nitrogen fixation, on biological nitrogen fixation. He got a great deal of funding from the Bill and Melinda Gates Foundation, very rightly so, to look at the engineering of cereals for that purpose. And it's a subject that has taken him for his main career to the John Innes, but he's just very, very recently moved from Norwich campus of the John Innes to the University of Cambridge, to the Sainsbury Plant Science Laboratory. I think it's wonderful that he's joining us now, because he's only just really moved lab. That's a very onerous process, as we can all imagine, if we don't know. He has many, many measures of esteem, but I think the main one is that he's in incredible demand for speaking at meetings, and it's wonderful that we were able to capture him today. So please give a wonderful round of applause and welcome to Giles Oldroyd.
Thank you, John. That was a very generous introduction. Okay, so it's a real pleasure, and thank you to Maurice for inviting me to this conference. It's my first visit to Saskatoon, but not my first visit to Canada, but it's always a pleasure to be back. So I'm going to talk today about biological nitrogen fixation and its potential for addressing issues of food security. So, if we go to the very intrinsic functions of plants, then at the core, it's about taking up nutrients, acquiring nutrients. So they're obviously very good at capturing carbon from photosynthesis, but the availability of nitrogen and phosphorus is often a limiting factor for plant production or plant growth. And as a result, in agriculture, we are using very large amounts of those those nutrients in fertilizers to drive food production. We heard already quite a bit about this earlier in the introduction. So we're at a point in our global food production where we really can't afford to stop using fertilizers. The impacts on the global population would be enormous if we were to cut down on the use of fertilizers. But one problem with fertilizer use is that it doesn't all end up in the plant. So it's the primary source of pollution. So the loss of nutrients through agricultural runoff is a real issue. And you see the eutrophication of our aquatic systems as a result of that agricultural runoff. And in some ways, you can see that as an environmental issue, but it's really an inefficiency in the system. In most industrial processes, you just wouldn't accept that those very valuable nutrients are not actually getting to where they want to be, or where we want them to be, but are actually ending up in the environment. And that represents a very significant inefficiency in the system. So what are other alternatives to using nitrogen fertilizers, and are there more efficient ways of acquiring those nutrients? Well, if we look out in the, sorry, I'm ahead of myself. So for a smallholder farming system in sub-Saharan Africa, They really deal with the opposite problem, though they do not have the financial resources to acquire fertilizers, or if they do, they're using very, very small amounts of nitrogen and phosphorus. And as a result, their crop yields suffer very significantly. So most smallholder farmers are achieving about 20% of the potential yields for that climatic region and for those soils. So obviously bringing fertilizers into those production systems is one way to address that. But to do so, you really have to address the infrastructure problems that most sub-Saharan African countries deal with. Really, the big problem with fertilizer is a very heavy, bulky commodity, and you have to get it from the ports where it's delivered through to the farmers in very isolated rural areas, and the transport infrastructure is simply not there to get those fertilizers to those farmers in a cost-effective manner. Now, in the natural world, most plants turn to microorganisms when they're in need of nutrients. So plants have acquired the ability to capture these limiting nutrients, phosphorus and nitrogen, through associations with microorganisms. And most plants have the capability to form symbiotic associations with arbuscular mycorrhizal fungi that heavily colonize the root and then ramify out into the soil. They're particularly good at capturing phosphate because they mobilize phosphate, they solubilize phosphate through extensive release of organic acids. But in many ways, you can think of the fungus as really an extension of the root surface area, really ramifying out into the soil and taking up the nutrients from the soil. Now, a smaller group of plants have also acquired the ability to associate with nitrogen-fixing bacteria that results in the formation of these nodules on the roots of the plant. And the primary among those species of plants are the legumes. And in those cases, those plants have the access to what is really an unlimited supply of nitrogen, the atmospheric nitrogen, which they can acquire through this interaction with nitrogen-fixing bacteria that use the enzyme nitrogenase to convert molecular dinitrogen into ammonia. So legumes have that capability. They form both the mycorrhizal and the rhizobial nitrogen-fixing symbiosis. And in contrast to our cereal crops, which are really a primary target for fertilization, they can form the arbuscular mycorrhizal symbiosis, but they lack any capability to associate with nitrogen-fixing bacteria. And in my research program, I'm particularly interested in advancing our understanding of how these symbiotic processes work in plants, and then using that knowledge to transfer the nitrogen-fixing symbiosis outside of the legumes, in particular to cereal crops, to drive the ability of those crops to acquire their nitrogen, not from nitrogenous fertilizers, but now from atmospheric nitrogen, which is a far more sustainable and equitable source of nitrogen than inorganic fertilizers. So in my talk today, we're running an engineering program in cereals, but I'm not actually going to talk too much about the engineering. What I'm going to tell you about and show you the evidence for is our knowledge of this process in legumes that is really informing us on how we approach this issue in the engineering of nitrogen-fixing cereals. But before I get there, I just wanted to mock up this graph, which is really the response of maize to fertilizer, to application of nitrogen. So we know that maize, or cereals in general, respond very well to fertilizer applications. And at the bottom of that graph, you can see smallholder farmers. They're applying fertilizers, or they're applying nitrogen in the range of 3 to 10 kilograms per hectare. And that compares to most developed world application of nitrogen, which is in the range of about 100 to 200 kilograms of nitrogen, depending on the country and on the crop. Now, most legumes fix nitrogen within that range, so they're fixing nitrogen around 120 kilograms of nitrogen. Optimally, it can go up to about 200. So if I want to replace nitrogen fertilizers, you really have to get up to those levels of nitrogen fixation that you see in the legume crop. But actually, for the smallholder farmer who's using only 3 to 10 kilograms of nitrogen, in fact, if you just increase that up to about 50 kilograms of nitrogen, you're already looking at doubling their yields. Now, so relatively, as far as a legume is concerned, relatively modest levels of nitrogen fixation have transformative potential for smallholder farmers in Africa. So, of course, I'm aspiring to put a pea nodule on a maize root, but actually, along the way, I can get a pretty crappy nodule or a very inefficient process and still have some pretty transformative potential. And so the big challenge for me, and one of the many criticisms I get, is how on earth are you going to put something as complex as a nitrogen-fixing nodule on a maize root? And I hope in my talk today you can recognize that actually I don't think it's as difficult as many people think. But secondly, that we can get a long way towards changing or improving productivity in Africa by what is actually a relatively inefficient nitrogen-fixing symbiosis in cereals. So if I can get 50 kilograms of fixed nitrogen into maize, I'll be extremely happy, and I think it's got a lot of potential for smallholder farmers. Okay, so we've been working on this field. I've been working in this field for nearly 20 years now, and we've been doing genetic dissection to understand how these processes worked. And through that genetic dissection, we identified this signaling pathway. And all the genes here are here because if you mutate them, you abolish the process of nodulation. And all of that genetic dissection that's performed in my lab and other labs around the world created this signal transduction pathway, which was really involved in recognizing the signal, nod factor, that's produced by nitrogen-fixing bacteria, and then transducing that signal through these calcium oscillations into gene expression changes that drive the process of nodulation. Now, one of the very early discoveries in this process that was really informative, I think, certainly for me in designing the engineering process, was that signal transduction pathway is not only required in legumes to support nodulation and the nitrogen-fixing symbiosis, but it's also required to support the arbuscular mycorrhizal association. So we have what we call a common symbiosis signaling pathway, which has a dual functionality. Now, what I'm drawing here is my previous, this is how I viewed this signaling pathway when I started the engineering program funded by the Bill and Melinda Gates Foundation. We were thinking of it as a bifunctional signaling pathway where you had two signals, nod factor from the nitrogen-fixing bacteria and MIC factor from mycorrhizal fungi, a core signaling process that was duly required, and then a bifurcation at the end that went into supporting either the mycorrhizal or the fungal symbiosis or driving nodulation in the rhizobia. And the reason we came to those conclusions was that the genetics was implying this. So for instance, all the components in gray were absolutely required for the nitrogen-fixing symbiosis, but were not required for the mycorrhizal association, and the components in black were duly required. So this is how our thinking was affected by the genetics. So importantly here, and if there's any message you take home from my talk today, I want you to take home the fact that this process, the process of nitrogen fixation and nodulation, is evolving or piggybacking on this process of the arbuscular mycorrhizal association. And what's critical here is that when we look at the evolution of these processes, so in red are the nodulating species, in blue is the whole phylogeny of the land plants, going all the way from mosses up to angiosperms. And the mycorrhizal symbiosis has a very ancient origin in the evolution of land plants. It goes right back to the very, very earliest land plants that we know of that were emerging around 450 million years ago. These were liverwort-like species. They just have a thallus. They don't even have a root at the time, but they already had a fully established arbuscular mycorrhizal symbiosis. And it's thought that fungal association facilitated the transition of plants from aquatic systems to the terrestrial systems. So as a result, the arbuscular mycorrhizal symbiosis is really ubiquitous within the plant kingdom. We see it across all plants, from liverworts all the way through to angiosperms, and importantly, it's already present, as I said, in our cereal crops. Now, in contrast, nodulation is rather restricted. It's just in a group of plants in the eurosids, and therefore this evolution, this process of recruiting this signaling pathway, and this signaling pathway that we'd identified, it actually must have evolved first to establish the mycorrhizal association, and then was later recruited during the evolution of nodulation in the nitrogen-fixing species. So in essence, this signaling pathway must therefore already be there in cereals, and in fact it's already been demonstrated with genetics that if you mutate these same genes, you abolish mycorrhizal associations in rice. So while we had discovered this signaling process in legumes, in fact it's not a signaling process that's restricted to legumes. It actually appears to be ubiquitous across the whole plant kingdom, and very importantly there is already there in the cereal crops. So when I first approached the Bill and Melinda Gates Foundation about the possibility of starting this engineering in cereals, I used this as a foundation for the justification, because if I had to recreate the totality of that signaling process, that would be pretty challenging. But I don't actually have to do that. What we had to do was put in what I thought were the components which were in gray, which were the nodulation-specific components that allowed inputs and outputs from this signaling pathway to give a nodulation output. In fact, I was wrong. And I'm going to show you the evidence and the data that demonstrated, in fact, that the thinking at this early stage was actually perhaps taking the genetics too literally. But really, if you look on the MIC factor component, we really didn't understand how mycorrhizal fungi were activating this pathway. And so as part of the program, we set about trying to understand how mycorrhizal fungi were activating this pathway, because that was a foundation to our understanding of how we were to modify the signaling pathway in cereals to deliver the novel components associated with nodulation. So really, this signaling pathway's primary function in most plants, and certainly its primary function in evolution, was the recognition of mycorrhizal fungi, and this is a mycorrhizal fungus growing in close proximity to the plant root. And we know that the plant is already capable of recognizing that fungus from a distance. It must involve diffusible signals. And a number of good candidates had been proposed for what those diffusible signals were that were coming from the mycorrhizal fungus. So it was demonstrated that mycorrhizal fungi can produce lipoketoligosaccharides. These were already well known because they were the nod factors that were produced by nitrogen-fixing bacteria to activate the process of nodulation in legumes. And these are essentially a chitin backbone that has a lipid tail attached to them, and they appear to be only function as microbial signaling molecules that are produced by the microorganism to activate the symbiosis signaling pathway in the plant. Now, in addition, unlike bacteria, fungi also make simple chitooligosaccharides, and the reason they do that is because chitin is the fungal cell wall structure. So in many ways, these short-chain chitooligosaccharides are a byproduct of having chitin as your cell wall. So fungi are producing both lipoketoligosaccharides, but also the more basic chitooligosaccharides. And we showed that both of these signals, the lipoketoligosaccharides and chitooligosaccharides, a simpler form, both can activate the symbiosis signaling pathway, and one of our primary outputs for that are these calcium oscillations that occur in the nuclear regions that are a hallmark of the symbiosis signaling pathway. Now, there does appear to be a qualitative difference between the way the chitooligosaccharides activate the pathway versus lipoketoligosaccharides, and you can see that just demonstrated in these traces. So LCO seem to activate the pathway more robustly. We get much more periodic and repetitive oscillations than we do when we use the chitooligosaccharides. Now, to understand the relative contributions of these signaling molecules in this process, we wanted to identify the receptors that were involved in that process. And we'd already identified through a collaboration with University of Aarhus, with the Jens Staugart's group, we'd already identified a number of receptor-like kinases in metacargo that were necessary for recognizing chitooligosaccharides and the activation of immunity signaling. And so this is just demonstrating that these two receptor-like kinases we call LIC9 and LIA4 were required for the perception of, this case, CO8, for activation of MAP kinases, and then the restriction of a fungal pathogen because of the activation of that immunity signaling. Now, we wanted to ask, having identified these receptors that were involved in perception of chitooligosaccharides, whether they were also involved in perception of chitooligosaccharides of symbiotic context. And indeed, that is the case. So this is just looking at in a number of mutants that are defective in different components of this signaling pathway. You can see that that the mutants DOMI1 and TOMI2, they sit at the core of the pathway, so that's demonstrating that symbiosis signaling pathway is required for this perception of chito-oligosaccharides. And then LIC9 and LIA4 that you can see at the bottom, these are the chitin receptors that are completely abolished in their capability to activate this calcium oscillation response. Now, NFP and LIC3 at the top of the panel are receptors that are involved in perception of lipoketoligosaccharides, and you can see that they're absolutely not required for perception of chito-oligosaccharides. So it seems we have two different receptor complexes, one that's recognizing LCOs and the other that's recognizing COs. If we look at the transcription profiling, essentially, it's a little hard to demonstrate this without a pointer, but the lipoketoligosaccharides are specifically recognized and require the LCO receptor for gene expression changes, and in contrast, the chito-oligosaccharides require the CO receptors for gene expression changes. And when we combine, if we take mutant, when we look at the function of this for the colonization by mycorrhizal fungi, so the LCO receptor, which is the NFP in that panel, it has no effect on mycorrhizal fungal colonization. But if we combine that with now the CO receptor, so that's the LIC9 NFP double mutant, we see a dramatic reduction in mycorrhizal colonization. So it appears that for a colonization by mycorrhizal fungi, you require both the CO and the LCO receptors that are dually involved in activating symbiosis signaling in response to mycorrhizal fungi. So in essence, we have mycorrhizal fungi producing two different signals, chito-oligosaccharides and lipochito-oligosaccharides. Both are capable of activating symbiosis signaling, but rhizobia, in contrast, are only making one of those signals, lipoketoligosaccharides, and only activating a single receptor complex to activate symbiosis signaling. And so when we look at the signaling pathway, this is where we thought we were in the beginning. We thought we had two different signals, and that was based on the fact that the genetics, the NFP and LIC3 that we knew were essential for recognition of nod factor, did not seem to be required for mycorrhizal colonization. But actually, in fact, the real situation appears to be that we have two different receptor complexes, one that's both that are capable of activating symbiosis signaling, but rhizobial bacteria are only activating one receptor complex, whereas mycorrhizal fungi are activating two receptor complexes. And because of that functional redundancy between those receptor complexes, it was hiding the fact that the LCO receptors were actually involved in mycorrhizal fungal colonization. Now, if we just go back to this model, we were spending an awful lot of time trying to engineer the nod factor receptors into cereals. But with this new understanding, it actually suggests that the LCO receptors were probably already present in cereals because cereals associate with mycorrhizal fungi. And indeed, that is the case. So I apologize, I don't know where these yellow lines came from, but it seems to be a default of transfer. So this is looking at barley, and you can see that barley is able to respond to both chitin COs and to lipoketoligosaccharides for activation of symbiosis signaling, and also the activation of gene expression associated with symbiosis. And importantly, the lipoketoligosaccharides I'm showing you there are not actually coming from mycorrhizal fungi. They're actually coming from nitrogen-fixing bacteria. So SM stands for Cynorhizobium meliloti, which is the nitrogen-fixing bacteria that associates with Medicago. And ML stands for Mesorhizobium loti, which is a symbiont of lotus. So the barley seems to be already capable of responding or perceiving the nod factor signals that are produced by rhizobial bacteria. And it's not the absence of that perception that is that differentiates a legume from a cereal. But if you actually look at then the activity of those signaling molecules, so on the top is Medicago, a legume, on the bottom is barley. And here we're just measuring the number of cells that respond with the calcium spiking response relative to the concentration of the signaling molecule. You can see that barley really doesn't differentiate between lipoketoligosaccharides, whether they're of fungal origin or bacterial origin, and doesn't really care which nitrogen-fixing bacteria they come from. In contrast, Medicago on the top there is exquisitely sensitive to one specific LCO, LCO, and that's the LCO that's produced by its nitrogen-fixing symbiont Cynorhizobium meliloti. So while cereals do actually perceive LCOs and are not, are actually perceiving LCOs produced by nitrogen-fixing bacteria, legumes have added on to that recognition a much more exquisite recognition, not just of an LCO molecule, but now an LCO molecule with a specific decoration. And in the case of Cynorhizobium meliloti, it makes a sulfated LCO, and that is the one that Medicago is so sensitized to. So although cereals can recognize the LCOs produced by rhizobia, it seems like in the evolution of nodulation, there does appear to be an add-on, an extra stringency of recognition that's been acquired by legumes for recognition of their symbiotic, the bacterial symbiont. However, what it does tell us is that it's not the recognition of these signaling molecules that differentiates a legume from a cereal. Cereals are actually recognizing nitrogen-fixing bacteria, and in that recognition, they're activating the symbiosis signaling pathway, which is the signaling pathway necessary for accommodation of these symbionts. So rather than so really it starts to come down to the question, well, what really differentiates a legume from a cereal? And the two processes I can say that definitely do happen in a legume that do not happen in a cereal. Firstly, legumes make nodules. So there's an organogenesis program that's activated in response to perception of lipoketo-oligosaccharides, and the result of that is the formation of these pink nodule structures on the roots of the host plant. And the other thing that legumes definitely do that cereals do not do is get colonized intracellularly by the nitrogen-fixing bacteria, and that occurs through this tube-like structure. This is a root hair cell, and the blue staining is the bacteria that are colonizing that root hair cell. And the plant makes this tubular infection structure that allows the bacteria to colonize the inside of the plant. Now, that tubular infection structure is very similar to when a fungus colonizes the plant. So a mycorrhizal fungus that's pushing its way or colonizing intracellularly through the plant cell will also form a tube-like infection thread, where the outside of the cell is the inside of that infection, and the fungus is pushing its way through that cell. So the actual capability to make this tube-like infection process, there's no evidence that we believe that is different to a fungal infection process. So it's not the capability to make this tube-like infection that's unique to legumes. It's the fact that legumes let bacteria activate that process and colonize the plant through that tube-like infection. But it's these two processes, nodule organogenesis and bacterial infection, that do seem to be the unique outputs that legumes have evolved that allow the process of nitrogen fixation. Now, I just want to come back to the evolution briefly and just tell you a couple of pieces of work that's just recently been published that I think are incredibly insightful into these two processes. So, as I said, nodulation is restricted to a relatively, or a group of angiosperms, the urosids. And if we take a closer look at that, this is now just a phylogenetic tree of all the species that were what are called the nitrogen-fixing clade. All the green lines are those species that associate with rhizobia, a single-cellular nitrogen-fixing bacteria, and primary among those are the legumes, but there's one group that's a non-legume, which is Parasponia and the Canabaceae, that can associate with rhizobia. And then all the red lines are nodulating species that associate with a filamentous nitrogen-fixing bacteria called Frankia. So back in the day, about 10 years ago, it was first discovered that all of these nitrogen-fixing plant species were clustered in this single clade called the nitrogen-fixing clade. It had been proposed that what happened at the base of that clade was a predisposition event that facilitated the evolution of nodulation, but that nodulation itself evolved on multiple independent occasions within that nitrogen-fixing clade. In many ways, the reasons that people proposed that was because all the gray bars are species that don't nodulate. And it was impossible to imagine that if you evolved the capability to fix nitrogen, why would you later lose it? And if it had been lost, it's been lost many, many times on many independent occasions. Well, two different research groups, essentially chose species within this nitrogen-fixing clade that nodulate and don't nodulate. They sequence the genomes of those plants and then ask for evidence for independent origins in the evolution of nodulation. And essentially what they found was that there was really no evidence in these different groups that there was independent evolution of nodulation. Rather, all those species that don't nodulate in this clade of species, or clade of plants, have lost the ability to nodulate. So so that suggests that there was a single origin in the evolution of nodulation within the nitrogen-fixing clade, and then many, many losses, which is an interesting thing to think about. Why would many, many plants lose the ability to fix nitrogen? It seems like a very beneficial process. Why would you lose that? But it also tells us that all species within the nitrogen-fixing clade once could fix nitrogen. So there's some very interesting crops in there. Apples, all the fruit trees, all your nut trees. There's hemp. There's walnuts, there's a lot of woody tree species. All of those were once nitrogen fixers. So the idea that we could actually re-engineer nitrogen fixation into those species is certainly probably much easier than in species outside the nitrogen fixing clade. But of all the independent losses that have happened in that nitrogen fixing clade, they've lost only 1 or 2 genes. 1 or 2 genes was lost in multiple independent losses of nodulation. In all these cases, they're losing nodulation, but they're maintaining the mycorrhizal symbiosis. So it points at there just being 2 genes that differentiate a nodulating species from a non-nodulating species within the nitrogen-fixing clade. Now, those 2 genes are 2 genes we already knew about, NIN, which is a key player in the emergence of nodule organogenesis, and RPG, which is a gene that's required for bacterial infection. So although this doesn't necessarily tell us that outside of the nitrogen-fixing clade those 2 genes are going to be all that we require, however, it does point at these 2 genes being really central to the nodulation process and specifically differentiating between mycorrhization, where it's been maintained, and nodulation that has been lost. So I'm just going to finish by telling you a little bit about NIN, because what we're focusing on right now is getting organogenesis working in a cereals and getting the activation of this root developmental program activated in response to symbiosis signaling. So if we look at where the nodule forms, it's initiated through cell divisions that occur in the inner root cortex, and those develop and divide from anticlinal divisions to periclinal divisions to form the nodule meristem that eventually emerges out of the root, pushing out through the root. Now, we thought for a while that this whole organogenesis program was really unique to legumes. We do not see the formation of any organs outside of legumes associated with symbiotic associations. However, Andrea Genre at the University of Turin just demonstrated very recently that actually mycorrhizal fungi do activate cell divisions. And here you can see the mycorrhizal fungus highlighted in red, and many cell layers down you see the recently divided cells that are activated in response to that mycorrhizal fungus. Now in this case, those cell divisions don't progress beyond that stage. So in a nodule process, you have the very first cell divisions exactly in that same cell types, but those cell divisions progress and progress ultimately to form the nodule meristem, whereas in the mycorrhizal association, we just get the first few cell divisions and it doesn't go beyond that point. However, what that's telling us is that this capability to associate to form a nodule probably evolved out of this pre-existing capability to form these cell divisions in response to mycorrhizal fungi. And so even there, something as what appears to be completely unique to the legume, the organogenesis program, it seems to be already derived from a pre-existing state where the plant is already activating cell divisions in the same cell types associated with mycorrhizal fungi, and then in legumes what they're doing is amplifying that process to drive cell division much further. Now, the components that we believe are involved in this amplification it appears to be this NIN transcription factor. I'm going to tell you a little bit more about that. So down in the inner cortex pericycle cells, we know that cytokinin is absolutely essential for the process of initiating a nodule, and that cytokinin drives the cell divisions that cause the nodule organogenesis. Now, to better understand this whole process, we took a very detailed RNA-seq approach where we're dissecting specific regions that were spot inoculated with bacteria. And at the bottom, you can see the very earliest stages of cell division, where at 12 hours, we see the cell cycle being activated. And this is a reporter that indicates cell division has been initiated. So you obviously get it with these RNA-seq approaches, you get a whole load of gene expression changes. We can put those into a temporal pattern. But if we look for where the cytokinin responses are happening, so that's the response regulators, these are the reporters of cytokinin, they're activated about 8 or 10 hours after the treatment with rhizobial bacteria, and that precedes by about 4 to 6 hours the formation of cell divisions. And this activation of cytokinin signaling is happening just after the induction of the expression of the NIN transcription factor. So the cytokinin, the absolute importance of cytokinin for this process was really highlighted when we looked at the cytokinin receptor mutant, CRE1, and there it abolishes 97% of the gene expression changes that are induced by rhizobia at 24 hours. So really, all this nodulation process is rhizobial bacteria activating the symbiosis signaling pathway, which is then activating the hormonal signaling pathway, cytokinin signaling, that then drives all the developmental changes that are happening associated with the nodule. And the transcriptome, those transcriptional dependencies on CRE1 were very heavily overlapping with the transcriptional dependencies on NIN. And this was pointing at NIN, this key transcription factor, having a function in cytokinin signaling. And indeed, if we just treat the plants only with cytokinin, that's the BAP, that's a synthetic cytokinin, and do so in both wild type and the NIN mutant, you can see that in the NIN mutant we consistently see a much reduced cytokinin responsiveness to this treatment with cytokinin. So you can see even for something like a response regulator, which is a specific marker for cytokinin, we see a dependence on NIN for cytokinin activation of that response regulator. Now, it's not an absolute requirement, so there's still some induction of the response regulator to cytokinin, but the total level of that induction is greatly reduced. And indeed, if we look at these reporters for cell divisions, so this is treating the plant with cytokinin, you can see that just cytokinin treatment activates a cell cycle in all of the cortical cells of the root, and that absolutely does not happen in the NIN mutant. So NIN appears to be amplifying this cytokinin responsiveness and driving cytokinin response into cell division. And indeed, among the genes that cytokinin is activating that are very dependent on the NIN are these two yuccas, which you can see in the middle of the panel there, yucca 2 and yucca 5. What the yuccas are is a rate-limiting step to auxin biosynthesis, and we believe that what the cytokinin is primarily doing is activating auxin biosynthesis, which then drives cell divisions for the formation of the nodule. And if we look at where those yuccas are expressed, this is promoter Gus of the yucca 2 and yucca 8. You can see that they're initiated below the sites of bacterial infection. You can see the infection threads forming in those roots, and below the site of that infection, right at the point where the cell divisions are initiated, we see the induction of these yuccas driving auxin biosynthesis, which then supports all the cell division changes that are necessary for formation of the nodule. So, in essence, what we think is happening is that the symbiosis signaling pathway is activating an organogenesis program that drives cell divisions in the inner cortex to form the nodule. But whereas we previously thought that all of that organogenesis program was restricted to legumes, in fact, what we and others have demonstrated is that is actually evolving already out of the mycorrhizal association, where mycorrhizal fungi are activating cell divisions in the inner cortex, but those cell divisions stay as they are. Whereas in the nodulation program, we see a massive amplification of that program, such that instead of just a few anticlinal cell divisions in the inner cortical cells, that is amplified and promoted through amplifying cytokinin signaling that drives the system much harder. So rather than a few cell divisions, you get many, many cell divisions going towards a formation of a meristem. Now, if I can take I was trying to think of a good analogy for how this is working. So, mum and dad get in the car and drive down the local freeway. They don't like to drive very fast, so they tend to drive quite slow. They're countering each other. Maybe mum is a chito-oligosaccharide receptor, dad is the lipochito-oligosaccharide receptor, both driving the pathway but not driving it very hard. So they drive it a little bit, they drive for half an hour, they get to the local shopping mall, and they stop and have a coffee, have a bite, a bit of lunch, and then go home. When they get home, their teenage son borrows the car. And he does not like to drive very slowly. So he drives down the freeway for around 100 miles an hour, likes to drive as fast as he possibly can, and he whizzes past the shopping mall where his parents stop for lunch. He whizzes past, he goes all the way to the big city of Saskatoon, and then he parties the night away with his mates, taking whatever his mates are taking, and trundles back home at some point in the late hours of the morning. Now, those two different outcomes look very different. So the lunch at the local shopping mall versus all-night party in the city, they look very different. But to get there, they use exactly the same car, they even use the same road. It's just the level of acceleration that was put into the system that that defined how far they got within that half an hour drive. And I think it's a very similar analogy here, whereby the mycorrhizal fungi are just activating the system, tweaking the system just to get a few cell divisions, whereas a nodulation system is pushing it very hard. And I think that the strength of the push is probably associated with that exquisite sensitivity to the lipochito-oligosaccharide receptors, or those signals that is happening in the legumes, that can drive the system very hard and push all the way to organogenesis and the formation of a nodule. Now, from an engineering perspective, that's a big difference. If I have to build the car and build the highway and put all of that into maize, that's a pretty challenging process. But we don't believe that's the case, because in fact, it's this whole process of nodulation is not coming out of the blue. It's utilizing all of these pre-existing processes, not just the signaling processes, but even the primitive states of the organogenesis program is already there and coherent and present in cereal crops. And if all I have to do is drive the accelerator of the system, that's a much easier engineering strategy than having to create the whole system in the first place. And I believe that certainly one of those accelerators is an in-transcription factor that seems to be driving the organogenesis program very hard to drive the formation of a nodule. So with that, I'd just like to acknowledge some of the people who have done the work. Two postdocs in my group, Fong Fong and Zhong Ho, did the early work that I talked about the CO and LCO receptors in cereals and in legumes. And then two postdocs, Katerina and Jody, and a graduate student, Yanis, have been working on the auxin work that I talked about later. And I should also acknowledge my collaborators, Jens Staugart, Simona Radutu at the University of Aarhus, and of course my funding from the Bill and Melinda Gates Foundation, and also with co-funding from BBSRC. Thank you very much.